Hello? Hello? Hello, everybody. I am Dr. Aissa Karima, and I am an assistant professor and a director of research at the College of Dental Medicine at Lincoln Memorial University. Today, I will be talking about my research project pertaining to nanoparticle targeted delivery for alleviating radiation-induced brain-blood barrier disruption as well as cognitive impairment. Okay, so it has been shown that cancer patients who have undergone radiation therapy have the higher prevalence for cardiovascular and neurological defect. This is independently of the cancer itself. This study specifically published in Lancet in 2017 showed that there is a significant increase in these patients 10 years or later of neurological defects as well as cardiovascular diseases. These diseases are highest in the aging population, and this shows some signs of early aging. So when we talk about cancer or cognitive impairment, that we are specifically looking into decreased verbal memory, spatial memory, attention, and novel problem-solving abilities. This has been shown, for example, in this study, after a whole brain radiation therapy, few months up to a few years following radiation therapy. So what does it mean? When you look at it, when you look at the brain or the brain-blood barrier specifically, you can see that the whole brain-blood barrier function relies on these monolayer of endothelial cells, or endothelium. So once we have the disruption of this barrier, we can say clearly that there is an increase or a breakdown of the brain-blood barrier. So if you look at the graph into the right of the slide, you can see when you have a breakdown of the brain-blood barrier, what follows later is literally toxicity to neurons. So either you have blood components coming directly in contact with these neurons, leading to the death, or you have a formation of the thrombus, which lead to a blood clot, and then some sort of impairment or deficiency in oxygen supply into the neurons that leads to death. So at the cellular level, in other publication as well as our lab, you can say clearly that the first target for radiation therapy is DNA damage. However, what we have seen at the acute level and the chronic level is that mitochondria, which is an organelle in these cells, can be targeted as well. So when we have mitochondria targeted or dysfunction, this will lead to an increase of mitochondrial ROS production. That mitochondrial ROS production could leak into the cytosol and lead ultimately to more DNA damage. However, mitochondrial DNA damage could be also one of the other aspects that we have seen and we have shown in our lab that is existing. So what happens when we have these mitochondrial DNA damage in the mitochondria following radiation therapy? Is what we need to know, at least at this point, that mitochondrial DNA is very similar to the bacterial DNA. And in that aspect, it has this ability to actually target and activate the toll-like receptor 9, or TLR9. Once this mitochondrial DNA is getting out of the mitochondria to the cytosol or to the extracellular space, that will be considered as a damage-associated molecular pattern, or DAM. So for the purpose of this presentation, moving forward, I will refer to mitochondrial DNA as a mitochondrial dam. So once we have this mitochondrial dam activate toll-like receptor, that will lead to inflammatory cascades that leads to a status of inflammation. If this status of inflammation is chronic and perdure during the time, that will lead to a more ROS production and more mitochondrial DNA damage and more mitochondrial DAMS release. So based on this aspect, we hypothesize that cranial radiation therapy will lead to release of mitochondrial DAMs, which in a feed-forward mechanism will lead to more TLR9 activation, more ROS production that will lead to a stressful event for the cell, leading to brain-blood barrier disruption or permeability, as well as cognitive decline. So to test this hypothesis, we wanted to make sure, confirm first in our studies or in our model, that we do have a mitochondrial ROS protection, mitochondrial DNA damage, and mitochondrial DAMS release. So we have assessed this in 2 different models. We did that in vitro in human brain microvascular endothelial cells, as well as C57 black cyst mice. So as you can see here in this graph, upper left is mitochondrial ROS production is much higher following radiation therapy, and you can see this at 24 as well as in 10 days. When we applied a mitochondrial ROS scavenger in these cells, you can see that there is a significant decrease, if not complete blockage, of mitochondrial ROS production following radiation therapy. In terms of mitochondrial DNA damage, we do see the same pattern. You can see there is an increase of lesions in mitochondrial DNA following radiation therapy. You can see this at 24 as well as in 10 days. However, when you apply the MTT, which is a mitochondrial ROS scavenger, you see much less, if none, of the mitochondrial damage. In terms of mitochondrial DAMS release, we look at the supernatant from human brain macrovascular endothelial cells 10 days post-radiation, whether there is a release of these fragments, and you can see that there is much higher fragments content in the supernatant from irradiated cells. However, if we have prevented or treated these cells with a mitochondrial ROS scavenger, we did block this completely. Same thing happened in the serum for C57 black cyst mice that have been subjected to radiation at 1 day post 1 day and post 10 days. As you can see, this is just the control, and then you see that there is a radiated C57 24, irradiated C57 at 10 days, and there is a higher number of or levels of these mitochondrial DAMs. And when we pre-treated these mice with a mitochondrial scavenger, we did reduce this significantly. This shows that when we have radiation therapy, we have an increase of mitochondrial ROS, we have an increase of mitochondrial DNA damage, and this ultimately leads to an increase of release of these mitochondrial DAMs. So next, we look at the role of toll-like receptors. Since mitochondrial DAMs are known to activate TLR9 receptors, so we made sure that this is also feasible in our project. As you can see that there is an increase, always an increase of mitochondrial ROS production following radiation therapy. And this is also shown when we have mitochondrial DAMs just by themselves applied to the cells, as well as when we use a TLR9 agonist. However, in terms of radiation therapy, when we applied antagonists for TLR9 and we subjected the cells to radiation therapy, as you can see, there is a significant decrease of mitochondrial ROS production in these cells following radiation therapy. The same pattern has been shown for mitochondrial DNA damage. So more ROS you have, more damage of mitochondrial DNA there is. And if you block that ROS, you have less, of course, damage. Of course, we use these as positive controls. Mitochondrial DAMs as TLR9 agonists have this ability to induce mitochondrial DNA. Again, we confirm that radiation therapy will lead ultimately to toll-like receptor activation. When we block this toll-like receptor activation, we might block these events that have been shown in radiation therapy. So we made another set of experiments to confirm that there is a correlation between radiation therapy and brain-blood barrier disruption. So in vitro, we use the permeability as well as a transmembrane resistance as a way to assess that. As you can see, there is more permeability on the endothelial cells when you have radiation therapy. It is much reduced when you have an antagonist TLR9 present in these cells. Same thing for resistance. You have higher resistance when you actually block the toll-like receptor following radiation therapy. In vivo, we did test the same thing, but we sectioned the brain following radiation therapy, and we stained for blood vessels in green, as well as extravascular leakage in red. As you can see, there is much more leakage following 24 hours radiation therapy in these mice. And when we actually used an antagonist for toll-like receptor following radiation therapy, as you can see, there is almost no leakage. So again, we confirmed that there is the link between radiation therapy, TLR9 activation, and endothelial barrier disruption. So we wanted to make sure we have a mean to target specifically endothelial cells that have been subjected to radiation, and to make sure also we deliver the toll-like receptor uniquely to these cells. Otherwise, we will have to block on the toll-like receptor everywhere, which is now defies the purpose of this study. So to do that, we have used nanoparticles coated with cyclic RGD. Why cyclic RGD? It has been shown that once you have a stress such as radiation therapy stress onto cells, on the endothelial cells, these cells will express these integrins alpha-5, beta-3. So the component, the peptide RGD, cyclic RGD, has been shown to bind efficiently to these integrins, So it has been shown that it's also able to go to the brain, as you can see here in this paper, and it has the ability to concentrate to the brain. So we used this technique and made sure first of all, when we have irradiation, our cells have the ability to actually express these on the integrins. As you can see here, we have a Western blot following radiation therapy. We have an increased protein production or protein expression of these integrin alpha-5 and beta-3. And it's also quantified, as you can see here. Also, we made sure that the cells have the ability to uptake and translocate these cyclic RGD and nanoparticles into the cell. As you can see here, we irradiated cells and subjected them to a cyclic RGD-coated nanoparticle, and we see an increase of uptake following radiation therapy in these cells. So first, we did confirm that we can use cyclic RGD nanoparticles as a mean to deliver and target specifically the irradiated cells. So we confirmed that this technology can work in our model. As you can see, we used an ODN 2088, which is a TLR9 antagonist, as well as nanoparticles, which are alone, which are not loaded with anything. As you can see, when we use the nanoparticle alone, we did not prevent the mitochondrial loss production. However, when we use the ODN loaded cells, we did prevent significantly the mitochondrial loss production in these cells. So that we can confirm that radiation therapy does induce mitochondrial loss production, but when we block the mitochondrial loss production or we block the toll-like receptor, we actually can block this loop. Okay, so now we made sure that we were able to have the cyclic RGD nanoparticle go to the brain and be taken by the endothelial cells that have been irradiated. So same thing, we used 57 black cyst mice, and we irradiated only one half of the brain, and the other half was used as a control. As you can see here, the non-irradiated hemisphere did not show any cyclic RGD-coated cells. Oh, yes, nanoparticles. However, here you can see significantly a green, which is a coumarin 6, which is coding the cyclic RGD nanoparticles. It is lining the endothelial cells, which shows again that there is a specific binding of these cyclic RGD nanoparticles into the irradiated endothelial cells in the brain, as well as their uptake into the cells. So we use this technology again in vivo in the 57 Black-Cyst mice. And again, we used mice that were half hemisphere or half brain irradiated to use the other half as a control. So in the nanoparticle alone, in this left side, you can see that there is a leakage. You can see that there is a red fluorescence indicating there is an extravascular leakage. However, when we use the nanoparticles that are loaded with TLR9 antagonist, we did almost not see any leakage. When we quantified this data or these images, you can see that with a nanoparticle alone, we do see an increase of leakage, which is indicated by here IgG. And once we have used the TLR9 antagonist, we blocked completely the leakage following radiation therapy. In these data, we confirmed that radiation therapy does go through TLR9 activation to induce endothelial barrier disruption. Okay, so the next step in our study, or the last step in our study, is to confirm that radiation therapy will lead to cognitive decline. As you can see, we have used the novel object recognition test, which is basically testing the ability of the cognitive function of mice before and after radiation. So the discrimination index indicates if it is higher, indicates a normal cognitive function. If it is lower, it indicates there's some sort of impairment. As you can see, at 10 days, 6 months, as well as 1 year, we see a complete decrease of discrimination index in these mice following radiation therapy. So we test this in our mice following nanoparticle loaded with a TLR9 antagonist. And as you can see, we have a higher, much higher discrimination index in the loaded nanoparticles with the drug compared to the empty nanoparticles, which are not loaded. As you can see that the toll-like receptor blockage did help in preserving the cognitive function following radiation therapy. So again, we showed that the radiation therapy does go through the toll-like receptor activation to induce a cognitive dysfunction. So in summary, we have shown in this project that radiation therapy, we did confirm, that radiation therapy does induce an excess of ROS production, mitochondrial ROS production, mitochondrial DNA damage. This has been already shown in our lab in other papers. However, what was not shown is that there is cell-free mitochondrial DNA fragments, which are damage-associated molecular patterns, have the ability to activate toll-like receptor, which in a feed-forward mechanism induces more ROS and more mitochondrial DNA damage, and a sustained effect of a stress or mitochondrial dysfunction that could lead to brain-blood barrier disruption and cognitive dysfunction. So these brain-blood barrier breakdown and cognitive decline are shown to be related directly to toll-like receptor. When we blocked the toll-like receptor, we were able to prevent these consequences following radiation therapy. We believe that blocking the toll-like receptor can prevent the early aging that we have seen in cancer survivors that are directly related to radiation therapy or anti-cancer therapy. With that, I would like to thank all the people who were involved in this project and from my previous work, which is Dr. Grumbach lab at the University of Iowa, as well as my current lab and all our fundings. Thank you.